Okay, so this video is going to be all about how neurons communicate, and before I go into all the nitty-gritty details, let's start off with just a description of what's happening here. So you're seeing here basically a long chain of communication happening from one neuron to the next, and there's really just two forms of communication, two forms of signaling that are happening here. The first one happens at the synapse, which is, as you can hopefully see here, the axon terminals forming a close but not physical connection to the dendritic spines of the neuron cell body. And right here is where we're going to have neurotransmitters acting and then binding at their targets. And this is going to induce a long series of chemical processes that we'll talk about later within the neuron cell body that ultimately results in a nerve impulse being generated, or you could just think of that being an electrical signal that's going to travel down the axon and it's going to land at the axon termini or terminal buttons. There's like 3 million different words to describe the same thing. That's going to result in more neurotransmitters being released at the next neuron. And so it's this long chain of processes happening that result in that signaling cascade. And so that's what this video is going to be about. So the first thing that I really wanted to stress is the concept of a membrane potential. And let's just say that you're a very well-off biochemist or cell biologist, and you find some neurons that you've grown successfully in culture. I know, that's the best picture. And you zoom in on just, say, for example, this one area, and you really just you magnify that, and you get some little small devices that can measure the electricity in terms of millivoltage. There's your phospholipid bilayer. That's the outside of the cell. That's the inside of the cell. So you stick your little electrode on the outside, and you find that it, on the outside, has a positive formal charge. And on the inside, collectively, it has a negative formal charge. Now, for the most part, the places of the cell tend to be electrochemically neutral, right? But just in these certain areas, you find this thing called a resting membrane potential. And it is about negative 70 millivolts, and since it's negative, we tend to say that it is polarized. Okay, now there's a lot of variation in what that specific number is. It's always negative, but there's variation in what it is. So let's say that you measure it right here and it was negative 70. Let's say you measure it, I don't know, right here, it's negative 40. You measure it right here, it's negative 90. It varies depending on the region, the specific area of the cell that you're looking at, and the cell type. Okay, so all these numbers that we're talking about here, these are just approximations or averages. I don't know the details of it. But this negative membrane potential is maintained by the activity of a sodium-potassium ATPase pump. So it's a form of active transport. We're using ATP to actively move molecules in and out of the cell. And in this case, we're actively moving 3 molecules of sodium out of the cell. And then we're going to actively move 2 molecules of potassium into the cell. Now, I'm not going to stress the details of the mechanism behind it. There's lots of great animations that do a better job of explaining it than I could. That's a little bit outside of the context of this class, but just notice that if you're losing 3 positively charged cations and you're only gaining 2 positively charged cations in the end of it, then the net gain is going to be negative. The membrane potential is negative. I'm going to write this out here, that polarization, being in a polarized state, means that it has a negative charge. There are other things that can happen, though, that we can observe inside the cell. Sometimes we can observe depolarization happening, where the membrane potential gets reduced, or you could just think of it as becoming more positive, the same thing as being less negative. What's in a name? Or there's also hyperpolarization, which is where the membrane potential is going to increase and become more negative, because if polar means negative, then hyperpolar means more negative, like the extreme of negative. And those are two things that we see happening all the time at the cell body. It's going to be depolarizing and it's going to be hyperpolarizing. And the reason that cells do this is to generate things called graded potentials. Okay, so these are graded potentials, are known as short, localized changes in the membrane potential. Now, sometimes they can be depolarization, making the cell more positive, or they can be hyperpolarization, making the cell more negative. But what we found is that they get weaker as they travel. This is called a decremental signal. So you have some type of a stimuli here, and there's going to be a little bit of a spread of depolarization. I'm not going to stress the detailed mechanism of how that depolarization spreads, so much as you understand that, yes, there's this inversion of charge happening there. And it's graded, we call it graded because the magnitude of it varies with the stimuli strength. So the stronger the stimulus, and I'm being abstract here. When I say the word stimuli, it can be like we're seeing here, just literally a physical pressure there. It can be heat, it can be light. But in this context, for most of the motor neurons here, which is kind of what we're going to talk about first, it's chemicals called neurotransmitters, okay? So the strength depends on how much depolarization happens, and it gets weaker as it diffuses away from the point of stimuli, in this case, the point where the neurotransmitter bound, okay? When a graded potential is caused by a neurotransmitter, we call that a postsynaptic potential. So here, right here is a synapse. And this is right here after the synapse, we call that a postsynaptic potential, okay? And again, it can be a depolarization or hyperpolarization. Both of these are forms of graded potentials. Graded potentials happen at the body, right here. And they can lead to action potentials being generated at the axon. But I want to really stress the fact that graded potentials happen at the body, and the action potential is not generated at the body. The action potential is generated at the axon hillock, or specifically at the axon, okay? One of the things that throws a lot of people off is this positive and negative signaling here that you're seeing happen. What that is, if we were to just kind of represent this graphically, that inversion point there is a little blip going up in the charge of the membrane potential. That's all that is. For some reason, that inversion sign really throws people off. I don't know why. It's just the same way of representing a spike in the membrane potential. Okay, so I said that there were graded potentials are just forms of short localized changes in the membrane potential, but there's actually two types. There's excitatory and then there's inhibitory. Postsynaptic potentials is just a form of graded potential that happens at the cell body, and in this context we're talking mostly about neurotransmitters. Yes, there are other things that can cause it, heat, light, pressure, if you're a sensory neuron, but let's just imagine for the purposes of this that you're a motor neuron, and this is what we're talking about here. So, what happens when an excitatory postsynaptic potential, or a postsynaptic graded potential, if you want to think about it, binds, is it's going to cause a single type of chemically gated ion channels to open on the postsynaptic membrane at the cell body or at the dendrites, right? And this is really important here. The potassium is in K, and the sodium, they're both positively charged, but they're going to diffuse simultaneously through the membrane. And I don't know if you can see this picture, but basically we have sodium over here diffusing in, and then we have potassium diffusing out. Okay, that's the same mechanism for depolarization. So you might be wondering, well, okay, but both sodium and potassium have a positive +1 oxidation state. Why would that result in a net result of depolarization? And there's basically a lot of ways that you could think about it. I think about this as being basically the exact opposite of the sodium-potassium ATPase pump. Or you could just think about it in the fact that in any cell, the most abundant intracellular cation is potassium, and on the outside of the cell, the most abundant intracellular cation is sodium. And on the inside of the cell, we said that the resting membrane potential is -70 millivolts, anywhere around there. So what that means is that sodium's electrochemical gradient, so the desire that sodium has to go inside the cell in order to reach equilibrium, is a lot stronger than potassium's desire to leave the cell. Potassium just wants to leave the cell based off of concentration gradients, not really so much with charges. Okay, so yeah, potassium does leave. It doesn't leave in as large of a quantity as sodium does. And anyways, what this results is depolarization, so it's becoming more positive or less negative, depending on how you want to word it. You might be able to see here, there's this little increasing blimp there. And that's what we're happening here, is we're talking about excitatory postsynaptic potentials. And what this is that this can trigger an action potential at the axon hillock, not at the cell body. People get that thrown off all the time. It happens at the axon hillock. Now, the closer your synapse is to the axon hillock, the more efficient it's going to be. And when I say more efficient, I mean it's going to take less neurotransmitters just by number to bind there to initiate that action potential. Now, not all graded potentials are excitatory. Some graded potentials can be inhibitory, okay? So inhibitory postsynaptic potentials or inhibitory postsynaptic graded potentials cause hyperpolarization, okay? So we're gonna become more negative, and we're gonna reduce that neuron's ability, or reduce the chance that it's gonna be able to generate an action potential. And the way that they do this is kind of different in that potassium, something that's positively charged, I hope you can see that there, is going to leave. So we can either lose something that's positively charged, or we could have chlorine going in, something that's negatively charged going in. And either way, regardless of the mechanism, sometimes it's both, sometimes it's either or, is the net result is hyperpolarization. The cell becomes more negative and it's less likely to generate an action potential. So as I had previously mentioned, graded potentials may or may not lead to action potentials. It depends on the type, whether or not it's excitatory or inhibitory, but there's really just two mechanisms by which graded potentials send their signal: temporal summation and then spatial summation. So in temporal summation, temporal in this context, we're talking about time. This is where we have one or more presynaptic neuron transmitting neurotransmitters in very rapid bursts quickly, and it doesn't allow the membrane potential to fully reset, and that summate, producing a strong depolarization event. If you could imagine it kind of like tossing a balloon very frequently, you keep tossing up that balloon, it gets higher and higher and higher and higher and higher and higher and higher, and eventually you're going to have an action potential generated. So here we have just this one single synapse here, and it's going to, in rapid succession, release many, many, many excitatory or inhibitory neurotransmitters. And in this context, we'll say it's excitatory. That results in these spikes here. We're seeing these spikes here, and then that's the generation of the action potential, okay? I really want to stress the fact that these can be either excitatory or inhibitory. So if it's happening at one point in space and having in rapid bursts, we call that temporal. And that can be excitatory or inhibitory. I think I've said it like 14 times now. But anyways, spatial summation, we're talking about space here. This is where the postsynaptic neuron is stimulated at the exact same time by a large number of terminals from either the same or from different neurons. So right here would be an example of spatial summation, and this would be an example of spatial summation. This is an example of spatial summation with two excitatory postsynaptic potentials, so they're both going to converge to give this really rapid spike there. You think of it as both as converging on this one point that's going to result in an action potential being generated. Or you can have excitatory and inhibitory ones binding, both releasing their neurotransmitters at the same time. So the inhibitory, as we can see here in I, brings it down, and then the excitatory brings it up, and there's kind of this competing action going on between the two to either convince the axon hillock to fire an action potential or to convince the axon hillock to not fire the action potential.